Good morning, students. Today's topic is carbohydrates. Carbohydrates are the most abundant organic molecules in nature. Carbohydrates are also known as biomolecules. These carbohydrates break down to give energy, water, and carbon dioxide. Glucose is the primary carbohydrate our body uses to produce energy. Carbohydrates also serves as energy stores like glycogen in animals and starch in plants. Carbohydrates are also present in the cell wall of plants and microorganisms. Carbohydrates are intermediates in the biosynthesis of fats and proteins. Carbohydrates are also found on the surface of the cell where they act as road signs, allowing molecules to distinguish one cell from another. ABO blood markers found on RBCs are made up of carbohydrates. Carbohydrates in our body prevents blood clots. Carbohydrates are also found in genetic material like DNA and RNA. They are one of the three components of genetic material. Chemically, carbohydrates contains carbon, hydrogen and oxygen. The general empirical structure for carbohydrates is CH2ON. By viewing this formula, these can be called as hydrates of carbon. The functional group present in the carbohydrates are aldehyde, ketones and many hydroxy groups. So they can be defined as polyhydroxy, aldehyde and ketones. The building block of all carbohydrates are simple sugars called monosaccharides. Now, classification of carbohydrates. Carbohydrates can be broadly classified into sugars and non-sugars. Sugars are crystalline, water-soluble sweet substances like glucose and fructose. Non-sugars are amorphous, water-insoluble, tasteless substances, example starch and cellulose. Carbohydrates can also be classified on the position of the carbonyl group. If it is terminal, the functional group is aldehyde and the sugars are called aldose. And if the carbonyl group is present other than terminal position, the functional group is ketone and we call them as ketose. Now the classification on the basis of sugar molecules. If the carbohydrate contain 1 sugar molecules, we call them as monosaccharide, and if they contain 2 to 10 sugar molecules, we call them as oligosaccharides. And if the number of sugar molecules are more than 10, we call them as polysaccharides. Examples of monosaccharides are glucose, fructose, galactose, oligosaccharides, sucrose, lactose, and polysaccharides, starch, cellulose, etc. These monosaccharides can be further classified on the basis of number of carbon atoms. Like if the number of carbon atoms are 3, we call them as triose. 4 carbon atom, we will call them as tetrose, and 5 pentose and 6 hexose. Oligosaccharides are classified on the basis of number of sugar units. If the sugar units are 2, we call them as disaccharides, trisaccharides and tetrasaccharides. Polysaccharides are classified further like homopolysaccharides and heteropolysaccharides. Homopolysaccharides have similar monosaccharides repeating units, but heteropolysaccharides have different monosaccharides units or sugar molecules. Now, monosaccharides. Monosaccharides are the simplest group of carbohydrates and are often called simple sugars since they cannot be further hydrolyzed. Monosaccharides have sweet taste, and sweetest sugar found in nature is fructose. These are solid at room temperature and these are extremely soluble in water. The high solubility in water is due to large number of hydroxy groups present. These monosaccharides are insoluble in non-polar solvents. They possess free aldehyde and ketone group. This is their general formula. And this classification I have discussed. Now stereochemistry in monosaccharides. Monosaccharides have multiple chiral centers. A chiral center is a center or an atom which have 4 different atoms or group of atoms attached to it. If we take the example of glucose, it has 4 different chiral centers. each having tetrahedral geometry. Groups attached to each chiral centers have two different arrangements or mirror images, which results in stereoisomers. The number of stereoisomers for a molecule increases with increase in the number of chiral centers in a molecule. The general formula for determining number of stereoisomers is 2 raised to power n. In glucose, there are 4 chiral centers, so number of stereoisomers possible are 2 raised to power 4 equal to 16. This is glucose and this is fructose. In glucose, the number of chiral centers are 4. In fructose, the number of chiral centers are 3. Now, Fischer projection for representing the stereoisomers. This is the Fischer projection. In Fischer projection, the chiral carbon atom is not shown, but it is assumed to be present at the intersection of the two lines. Now, DL designation of sugars is relative configuration. All sugars having the OH on the chiral center farthest from the carbonyl group on the right side of the molecule are designated as D sugars by comparing with the glyceraldehyde in which OH group is present on the right side. And in all L sugars have the OH on the chiral carbon farthest from the carbonyl on the left hand side. This is the glyceraldehyde molecule. In G form, OH group is present on the right side, and in L it is present on the left hand side. All other sugar molecules are given DL-L configuration by comparing with the D-glyceraldehyde. In erythrose, there are 2 chiral centers, but we have to note the position of OH of the highest number stereo center. In this lower chiral center, OH group is present on the right hand side, so it is given capital D form. And in case of this molecule, L-erythrose, the OH group in the next molecule, OH group is present on the left hand side.